Hello everyone, my name is Dr. Deepika and today's topic of discussion is industrial production of citric acid. Citric acid was originally produced from lemons in 1784 by the Swedish chemist Carl Wilhelm Scheele. The discovery of its accumulation by Aspergillus niger in the early 1920s led to a rapid development of a fermentation process which 15 years later accounted for more than 95% of world's production of citric acid. Citric acid is usually occurring acid found primarily in several varieties of fruits and vegetables, with citrus fruits such as lemons and limes containing the highest amount of citric acid. The organic acid has many uses, including as a food additive or preservative, ingredient in cosmetic products, in pharmaceuticals, and as a powerful cleaving agent. It has pleasant taste and property of enhancing existing flavors have ensured dominance in industry. It is good natural preservative and is also used to add an acidic taste to food and soft drinks. Today, citric acid is produced in bulk amounts with an estimated worldwide production of 4 lakh tons per year, most of which is produced by fermentation with the fungus Aspergillus niger. Now let us see the definition of citric acid. Citric acid, also known as 2-hydroxy-1,2,3-propane tricarboxylic acid with the molecular formula of C6H8O7, is a weak organic tricarboxylic acid found in citrus fruits. Citrus fruits like lemons, oranges, tomatoes, beets, etc. are those fruits which contain sufficient amount citric acid and they are classified as acid fruits. Citric acid is excreted from the cells in response to unfavorable intracellular conditions caused by increased levels of tricarboxylic acids. It is a natural intermediate in Krebs cycle. A crucial prerequisite for overflow of citric acid from Aspergillus niger cells is therefore increased level of Krebs cycle intermediates caused by anaplerotic reactions. Anaplerotic reactions are chemical reactions that form intermediates of a metabolic pathway. Citric acid is naturally non-toxic due to its widespread presence. Now let us see the mechanism of formation of citric acid. Citric acid is principal organic acid produced in citric acid cycle. The biochemical pathway in formation of citric acid is shown in the image. When mycelium grows actively, citric acid is produced as an intermediate in citric acid cycle and is further diverted to growth promoting biosynthesis or release energy. Now let us see some uses of citric acid. Citric acid are used as preservative and flavoring agents in food and beverages such as soft drinks. They are used as an emulsifying agent in ice creams. They are an excellent chelating agent. Citric acid is used as an acidulant in gels, creams and liquids of all kinds. Acidulants are chemical compounds that confer a sour or acidic flavor to foods. Next slide shows the different sources of carbon that can be used in the production of citric acid. Sucrose, according to laboratory studies, is the best source of carbon among various tested organic substances, particularly sugars, in producing high yields of citric acid. It was also reported that sucrose concentration exceeding 15% should not be used, since the excess amount of sugar remained unconverted to citric acid. When a part of sucrose was substituted by fructose or glucose, it resulted in lower yields of citric acid than when controls containing sucrose alone were used. As we know, sucrose is a disaccharide composed of two monosaccharides, glucose and fructose. Therefore, precaution is to be taken while using sucrose to avoid partial hydrolysis. Under laboratory conditions, the highest yields of citric acid are secured provided sucrose has been passed through an ion exchange resin. This is done for demineralization, that is loss of minerals of the sugar juice. So it is important to get rid of these metal ions which can otherwise hinder the process of citric acid production. Substrates of high purity such as hydrolyzed starch, pure glucose, refined or raw sugar, purified and condensed beet or cane juice have high yields in submerged culture. Beet molasses is extensively used as a carbon substrate in the fungal production of citric acid on a commercial basis. Beet molasses require pretreatment since it contains excessive amount of trace metals. Therefore, ferrocyanide or ferricyanide may be added to the production medium before sterilization. The metals of the beet molasses form a complex with the added chemical agent, thus eliminating themselves as the precipitate. So in case of beet molasses also, there is a need to remove these trace elements which can otherwise hinder the process of citric acid production. Now let us see the influence of trace metals. The type and quantity of metallic ions, example iron and zinc, required for citric acid production from Aspergillus niger fermentation largely depend on the type of fungal strain to be used in the process. Iron ions in higher concentration than 1.5 mg per liter strongly affect cellular morphology by promoting unproductive filamentous mycelial growth form and giving no increase in citric acid yield. This means that at a higher concentration, iron is only promoting the growth of the fungus but not of the product. 1 microgram per liter of manganese could completely ruin the production yield and cause organisms' morphology to switch from microbial pellets, as you can see in the first image, which are also known as citric acid productive form, to unproductive filamentous growth as shown in the second image. So by this we can conclude that the morphology of fungus within the fermenter play a very important role in the production of citric acid. Fungal pellets are more efficient in producing the desired product rather than mycelial mat floating on the surface. The fermentation medium is first freed of trace elements, example manganese, zinc, iron or phosphates through ion exchange resins because the trace elements exert inhibition of citric acid formation above a critical level. Therefore, the presence of these trace elements beyond a certain limit is not suitable for the production of citric acid. Next important components are inorganic salts. Apart from the carbon, hydrogen and oxygen supplied by the added carbohydrate, the inorganic elements namely nitrogen, potassium, phosphorus, sulfur and magnesium are needed in the fermentation medium for citric acid production. It is highly essential to add ammonium nitrate, potassium dihydrogen phosphate, dipotassium phosphate or magnesium sulfate heptahydrate only in a minimum quantity, since the presence of these metallic salts beyond their limits affects the fermentation adversely. Low pH values have found to be the most favorable for citric acid production. The pH of the medium is initially adjusted to 3.4 to 3.5 with HCl. On spore germination, pH drops down to the range of 1.5 to 2. The use of HCl has been found superior to nitric, sulfuric and acetic acids in producing higher yields of citric acid. Generally, calcium carbonate is not added to the production medium to bring about the neutralization of citric acid produced because its presence favors contamination. Moreover, its absence favors high yields of citric acid and a shorter fermentation time. Low pH values in Aspergillus niger citric acid fermentation are desirable for the following reasons. First is sterilization of medium is more readily affected. Second, formation of citric acid is favored at low pH. Third, formation of oxalic acid is suppressed at low pH. Fourth, the danger of contamination is minimized at low pH. Next, the exact required temperature of incubation depends in part on the fungal strain and the fermentation conditions. However, a temperature range of 26 to 28 degrees Celsius for 7 to 10 days is considered to be satisfactory. Now let us have a look at different microorganisms used for citric acid production. Large number of microorganisms including bacteria, fungi and yeasts have been employed to produce citric acid. The main advantages of using these microorganisms are its ease of handling, its ability to ferment a variety of cheap raw materials and high yields. The table shows the important species of fungi, bacteria and yeast involved in the production of citric acid, and the most important among these being the Aspergillus niger. It is very important to choose a suitable fungal strain for producing citric acid successfully. Many fungi have been reported to possess the ability to produce citric acid. For example, Aspergillus niger, Aspergillus clavatus, Penicillium luteum, Penicillium citrinum, Paecilomyces divaricatum, Mucor piriformis, Ustulina vulgaris and other species of mucor have been employed to produce citric acid in the laboratory or on a commercial basis. But only Aspergillus niger strains among these fungal strains are used for the following reasons. They are efficient, that is high yielding strains. They possess fairly uniform biochemical properties. They produce a negligible or small amount of oxalic acid provided fermentation conditions, that is pH and salts, have been adjusted to favor the formation of citric acid. They can be easily cultivated. Now let us see the types of production processes involved in the production of citric acid. Fermentation is the most economical and widely used method for synthesis of citric acid. The industrial citric acid production can be carried in three different ways. First is surface fermentation. In this case, Aspergillus niger floats on the surface of liquid medium. Second is submerged fermentation. It is the process in which the fungal mycelium grows as fungal pellets throughout the liquid medium in a fermenter. Third is solid state fermentation. It is a Japanese process in which special strains of Aspergillus niger are used with a solid substrate such as sweet potato starch. Surface or submerged fermentation techniques dominate over solid state fermentation in preparing citric acid by extraction from various juices. Promising results were obtained in fed batch process and by continuous fermentation. Citric acid fermentation using immobilized Aspergillus niger cells on various kinds of carriers such as glass, polyurethane forms, entrapment in calcium alginate beads, polyacrylamide gels, agar, agarose, cellulose carriers, metal screens, and polyester felts have given promising results. These are the methods of immobilization of the spores of Aspergillus niger, giving sufficient production of citric acid during fermentations. Now let us discuss all these fermentation processes one by one in detail. First is surface fermentation. The fermentation is usually carried out in aluminium trays filled with liquid nutrient medium to a depth of between 50 to 200 cm. Molasses solution containing inorganic nitrogenous salts is used as fermentation medium. The pH is maintained at about 2. Spores of Aspergillus niger are now distributed over the surface of the trays, and sterile air serving both as an oxygen supply as well as a cooling aid is passed over them. The mycelium develops on the surface. The fermentation is completed within a period of 7 to 12 days at 30 degrees Celsius. The culture fluid is taken out from the bottom of the trays, leaving the mycelial mat of Aspergillus niger more or less undisturbed. Citric acid is recovered from the fluid by precipitation. Fresh medium is added into the trays for the next batch of fermentation. The diagram gives an idea of the surface fermentation. Raw materials are stored in storage tank from where they are prepared for fermentation. Preparation involves mixing and heating with water. The complex substrates undergo starch hydrolysis to release fermentable sugars. After preparation, the substrates are now distributed over the surface of the trays, which are then inoculated with the spores of Aspergillus niger. The main advantages of surface fermentation are the low power requirements of the process, the high reproducibility of the process due to its lower susceptibility to interference by trace metal ions, and variations in the dissolved oxygen tension, since it is a stationary process. The disadvantage is that it is an older and more labor-intensive version of fermentation for citric acid production. Second type of process is the submerged fermentation process. Now this process can be carried out using either fungi, that is Aspergillus niger, or yeast. In the first type, the fungus is grown dispersed through a liquid production medium. Fermentation is carried out in fermenters equipped with a mechanical agitator for mixing and a sparger for aeration. For inoculum preparation, the mold, that is Aspergillus niger spores, are produced under controlled aseptic conditions. They are used in specific quantities to seed the inoculum tank containing inoculum medium designed to develop small pellets, that is aggregates of fungal hyphae, as you can see in the image, of size 0.2 to 0.5 mm, having a firm and smooth surface. This striking morphology has been shown to be critical for attaining high yields by submerged fermentation. So for submerged fermentation, fungi pellets are developed first in inoculum medium before adding it into fermenter. As we have discussed earlier also, pellets are more efficient in giving high yields of citric acid, which is produced extracellularly rather than mycelial mats. After the inoculum has been prepared, it is then transferred to the production tank under aseptic conditions. In contrast to the composition of the inoculum medium, production medium contains constituents so as to favor citric acid production rather than growth. So inoculum medium and production medium are two different mediums. Inoculum medium favors the growth of fungi, whereas production medium favors the production of citric acid in a high concentration. High aeration rates producing much foam are necessary, and it is allowed to grow for about 5 to 14 days at 27 to 33 degrees Celsius. Samples are regularly withdrawn to determine the citric acid and sugar content, revealing the progress of the fermentation process. In addition to this, pH, dissolved oxygen and solids contents are also determined. Now, submerged fermentation can also be carried out using yeast. The belief that industrial production of citric acid could be possible only by Aspergillus niger was disapproved in about 1970. Citric acid production by yeast is exclusively carried out by submerged cultivation. The submerged fermentation process is desirable because of its higher efficacy due to higher susceptibility to automatization. Citric acid is produced using yeast Candida guillermondi, grown submerged in a medium containing either glucose or blackstrap molasses with an equivalent amount of sugar. Using certain strains of Candida lipolytica, hydrocarbons and alkanes have been used as raw materials in citric acid production. The main advantages of using yeasts are that yeasts are characterized by greater resistance to high substrate concentrations than fungi, with comparable conversion rates, and have greater tolerance to metal ions that allows the use of less refined substrates. Using yeasts also give a better process control due to their unicellular nature. With yeast fermentation, the duration of the fermentation period was much shorter than that of the submerged culture process involving Aspergillus niger. The next type of process are the solid state fermentations for production of citric acid. It is the simplest method for its production. The solid substrate like bran is soaked with water and steamed so as to get the water content of the mash around 65 to 70%. A bran mash is simply wheat or rice bran mixed with boiling water until it reaches the desired consistency and is allowed to steep for 15 minutes. Steeping is again a process of increasing the moisture content of the substrate. After cooling the substrate to 30 to 60 degrees Celsius, Aspergillus niger is inoculated. The pH of the substrate is about 5 to 5.5 and is spread in trays to a depth of 3 to 5 cm after inoculation. Growth can be accelerated by adding alpha amylase, although the fungus can hydrolyze starch with its own alpha amylase. This conversion of starch into fermentable sugar is also termed as saccharification. During the citric acid production, pH drops down to the value below 2. The solid state surface process takes 5 to 8 days, at the end of which the entire citric acid is extracted with hot water. Citric acid production reached to a maximum yield of 88 grams per kg dry matter when fermentation was carried out with cassava, having initial moisture of 62% at 26 degrees Celsius for 120 hours. Cassava is a root vegetable, as you can see in the image. Now, after fermentation is complete, first step is the separation of biomass, that is mycelia or cells, from fermentation broth by filtration. The liquid broth obtained, known as mother liquor, is now transferred to recovery process. Now, recovery can also be done by two methods. First is the precipitation technique. The filtered broth is treated with calcium hydroxide so that the relatively insoluble calcium citrate crystals will be precipitated from solution. The calcium citrate is filtered off, washed with water and treated with sulfuric acid to precipitate the calcium as the insoluble calcium sulfate and release the citric acid. This citric acid is subjected to the treatment with activated carbon. It is demineralized by successive passage through ion exchange beds, and then the purified solution is evaporated to the point of crystallization in a circulating granulator or in a circulating crystallizers. The crystals are removed by centrifugation. The remaining mother liquor, that is the liquid remaining after substance has crystallized out, is returned to the recovery stream. This is the only commercially viable process existing, but the common precipitation process for isolation of citric acid produces 2.5 tons of waste per ton of citric acid. It is therefore reasonable to look for other methods of citric acid recovery. Another method for recovery of citric acid is by solvent extraction. Solvent extraction is a possible alternative to the classical method. However, the solvents tend to extract some of the impurities contained in molasses-derived liquors. The advantage of the solvent extraction method is to avoid the use of lime and sulfuric acid and the containment problem of gypsum disposal. The solvent extraction can be performed by adding 100 parts tri-N-butyl phosphate and 5 or 30 parts N-butyl acetate or methyl isobutyl ketone, which are to be mixed with the filtrate. The solvent is then extracted with water at 70 to 90 degrees Celsius. Citric acid is further concentrated, decolorized or bleached and crystallized. After we have recovered the crystals of citric acid, they are finally dried and packaged. Now let us have a quick review on the process of citric acid production. First step is the selection and preparation of raw material by passing it through ion exchangers to remove trace elements which can interfere with the entire process of citric acid making. The substrate is now inoculated with the suitable microorganism to carry out fermentation. Fermentation can be done by any of the three processes. In surface fermentation, the spores of Aspergillus niger floats on the surface of liquid medium. In submerged fermentation, the fungal mycelium grows as fungal pellets throughout the liquid medium in a fermenter. This submerged fermentation is also suitable when using yeast for the production of citric acid. For solid state fermentation, solid substrate are used and inoculated with the Aspergillus niger. After fermentation is complete, the next step is separation of biomass, that is fungal mycelia or yeast cells, from the fermentation broth. Now the next steps are for the recovery of citric acid by precipitation techniques. The filtered broth is first treated with calcium hydroxide to remove insoluble calcium citrate crystals. These calcium citrate crystals are then washed with water and treated with sulfuric acid to release the citric acid. The citric acid is now treated with activated carbon. After that, it is demineralized by passing through the ion exchange beds. Further, they are purified by evaporation and crystallization to obtain citric acid crystals. These crystals are then removed by centrifugation. Now, after the crystals are obtained, citric acid is dried and packaged. With this, we wind up the topic of industrial production of citric acid. For any doubts or queries, you can contact me through the given email ID. Thank you.